So I'd now like to proceed with the formal business of this meeting. Following the conclusion of the meeting, there will be a presentation by CEO Dr. Taylor and Principal Investigator Dr. Barry Snow, and there will also be an opportunity for shareholders to ask questions about the company and its performance. I wish to advise that I will be calling a poll on all resolutions required to be voted on today. I will also confirm that I propose to vote those proxies left to my discretion as the Chairman in favour of all resolutions. Proxy voting received prior to the meeting will be shown on the screen for each resolution dealt with today. Voting exclusions. Resolutions 1 and 10 today are subject to the following voting exclusions. No votes may be cast on Resolution 1 by A member of the key management personnel, details of whose remuneration are included in the remuneration report, or B, a closely related party of such a member. However, if a person so described may cast a vote on the resolution if the person does so as proxy, appointed in writing that specifies how the proxy is to vote on the proposed resolution. The vote is not cast on behalf of a member of the key management personnel or related party, and the vote is cast by the Chairman as the nominated proxy for a person who is permitted to vote, with express authorisation given to the Chair to exercise the proxy, even if the resolution is connected directly or indirectly with the remuneration of a member of the key management personnel of the company. No votes may be cast on Resolution 10 by any person who may participate in the proposed issue of equity securities under Resolution 10. Any person who might obtain a benefit, accept a benefit solely in the capacity as a shareholder if Resolution 10 is passed, and any of his associates. All shareholders may vote on Resolutions 2 and 3. I'll now table the 2017 annual report. For consideration of the company's financial report and the reports of the directors and auditor for the year ended the 30th of June 2017. I now invite shareholders' comments and questions on the annual report and financial statements. Mr. Lynch and Mr. Stanley of BDO are available to answer any questions on the conduct of the audit and the audit report. Are there any questions on the financial accounts? I'll now introduce each resolution requiring a vote today. You will be asked to fill in a proxy form. Please note that you're only addressing resolutions 1, 2, 3 and 10. So your proxy card has a number of resolutions on it that were sent out with the notice of the meeting. We're now dealing with only resolutions 1, 2, 3 and 10. And when you sign, if you could also please sign your proxy in the bottom right hand corner. Proxy voting received prior to the meeting is shown on the screen to my right. The first resolution is the adoption of the remuneration report. Proxy voting received prior to the meeting is now shown on the screen behind me, which is there. Resolution 2 involves the re-election of Roy Alston as a director. Proxy voting received prior to the meeting is shown on the screen. The third resolution is the re-election of Bernard Tuck. Sorry, did I flip through that a bit too fast? And resolution 10 is the approval of the 10% placement facility. So I now declare the poll open for voting on these resolutions. A member of the company's share register, Computershare, will act as returning officer. All members and appointed proxies eligible to vote were given a voting card upon admission to the meeting. All other persons are ineligible to vote. If you believe that you've issued an incorrect card, please see one of the registration staff located at the end of the meeting room here. So we now ask shareholders to complete their voting cards by placing an X in the appropriate box, either for, against or abstain. Once you've marked your vote, it will be collected by the returning officer's staff. And remember, you're only voting on resolutions 1, 2, 3 and 10. So I'll just pause now till the voting cards are collected. All cards been handed in? Nobody holding any? Okay. I'll now declare the poll closed. As it will take some time for the poll results to be known, they'll be announced at the earlier of the completion of our addresses today or of our announcement to the ASX later today. I have no notice of any other business, so I'd like to thank you for your attendance. I will now close the formal part of the meeting and introduce Dr. Taylor to be followed by Dr. Barry Snow. Here you go, Ken.
Thanks, Roy. This will be a relatively short presentation focused on the trial results, the status of the new projects that we're looking at, and then I'll introduce you to Dr. Barry Snow, the principal investigator of our clinical trial, and leave plenty of time for questions for you at the end of that. Last Friday, LCT, like you, were informed of the Parkinson's disease trial result. It was unexpected and we were disappointed. The study was cutting edge, and to complete that study is a tribute to the patients themselves, to Barry, the principal investigator, and his staff, the LCT staff. To complete that as a double-blind study, that meant that we got the result on the same day that you actually did. Maintaining the double-blind meant it was a powerful study, and as we release the top-line results, but we do get time to digest all of the study. So as I mentioned, the first part of what I'll talk about is the follow-up on the outcome of the clinical trial we've just completed, NT cell and Parkinson's disease. I'll give you an update on other projects. All through the year, we have been looking for pipeline extension opportunities, and I'll give you an update on where we're at with that. I'll describe to you what next steps we are taking at LCT, and then, as I mentioned, I'll introduce you to Barry Snow, and he'll tell you a little bit about the trial, and then we'll move into question time. Now, the trial that we just completed, the trial was designed to address 3 questions raised by our Ministry of Health. And the reason that that was important was that if we could answer them from the trial data, then we would qualify for a conditional approval through a fast-track system that we had in this country. And we had done a lot of work to follow up on that, and as you know, we raised funds for the continuation so that we could move quickly, hopefully towards a market approval. The 3 questions raised were to define the efficacy and any placebo contribution. In all Parkinson's study, and Barry Snow may comment on this, there is a placebo contribution, and we needed a statistical significant difference between the placebo and the different doses of NT cell that were used in the study. That would enable us to define the optimal dose of NT cell for implantation into the putamen area to treat Parkinson's patients. And then that would also help define the initial target Parkinson's patients, what stage of the disease were they at where the response could be expected to be the best, and if you like, there was a cost-benefit ratio. Now, this is the data that we released. It is top line only, and that's the information that we received last week. What we're looking for here is a measurement of the neurological rating scale, and the subgroup is the motor examination in patients who have come off their symptomatic treatment the day before. So that is the primary clinical endpoint at 26 weeks for the study, and everything we do is defined by the clinical trial protocol. So we have to follow that, and that defines what the primary endpoint is and what the statistical differences that we're expected to see. Now, as you can see, we had the three groups. The placebo is the blue, and you can see that the blue line over the period of time does have some effect. But like the three treatment groups, the implantation of 40 capsules on both sides of the brain, the green group, coming down, the 80 capsules per each side of the brain, also showing something promising, and the high dose NT-cell at 120 capsules each side may be tracking the worst here. Now that high dose is the highest dose we could use. We do have an independent data safety monitoring board, and they confirmed that we were at the dose limit. In other words, they would not recommend that we could go further with a dose of that size. So we did know that we could not give a higher dose of NT-cell capsules if there was any reason to suspect that we had not dosed high enough. Now, as you can see, the feature that sticks out a mile is the standard error bars, and they indicate there's considerable variation in the response between patients. The consequence of that is that we don't get to statistical significance. So therefore, what that means is that the primary clinical endpoint of efficacy, as defined in the protocol at 26 weeks, we have not met that primary endpoint. The other primary endpoints related to safety, and we did meet them. So at least in this situation where if you're going to try to do some good for patients with a very nasty disease where there's no treatments, we haven't done any harm. So the implantation in NT-cell is, from this study, proved to be a safe product. Now, the clinical trial follow-up becomes very, very important. The groups 1, that's 40 capsules each side and 80 capsules each side, we'll have 1 year data for these patients in December, and it's our intent to analyse that. That would address the situation. Is 26 weeks too early a time to look for an agent that would stop nerve degeneration or maybe make nerves grow again? So we will get longer term data. We do have a trial extension protocol, so we will also be able to go out to 18 months and 2 years. So we do have a long-term follow-up process that can be followed, and that is according to an approved trial protocol. The individual patient data, as I mentioned, there's considerable variation, and therefore also what we're looking at is tracking individual patient data to see how does that compare in each group. Is the group mean that we work off heavily affected by an outlier, good or bad, bad effect, good effect? So we will find that out as well. What that will enable us to do is also compare the result to patients in our first trial. You may remember there were 4 patients there. 2 of the patients will come out to 3 years after implantation with 40 capsules on one side by the end of this year. 1 patient, the first patient, is out to 4 years. And we look at those as being individual data, if you like. So to understand the individual variations in our second trial will enable us to comment on, well, what's different between the first trial and the second trial. The goal of this analysis is to understand the results. If that suggests any address of the 3 questions raised by MedSafe, then we would discuss that with them, but that does depend on the data that we get. If that in turn would suggest we have to do something more, then that would be the first call on our current funds would be to persevere with that in the hope that we can drive to clinical significance and hopefully towards a product. So how much cash do we have? In our September ASX release, we had Australian dollars 6 million. We have since received rebates from Callaghan. We get 20% rebate on our spend from Callaghan Innovation and a tax credit. So currently, with that on mind, probably about now we are at the same amount. We're roughly Australian dollars 6 million is the funds that we do have. So the new projects and their status, we've divided them between two that we've followed up on. The cell-based projects look at a product like NT cell, but approaching different targets. We have made progress in looking for the appropriate partners to work with these two indications here. In doing that, we have to go into confidential document disclosure, if you like. So those are two that, if you like, with the result we've got, we can progress from quite an advanced stage to see if we can identify a worthwhile project for investment. The non-cell-based ones, we have followed the possibility of bringing in a CNS active chemical, and this would be one that has already synthesized a lot of data and ready for a clinical proof of principle study. So if we have the ability to do that, and New Zealand is a good place to do that, once again, it would depend on, is there an attractive investment project that gives us a return working with the provider of those compounds. Now, the project we have announced previously is the collaboration with the Centre for Brain Research here in Auckland, run by Sir Richard Fall. And what that's all about is trying to identify what you would call a pericyte protective agent, abbreviated to PPA. So what are pericytes? These cells surround blood vessels, capillaries, in all of our organs. They intertwine with the epithelial layers to, if you like, maintain the integrity of the blood vessel, if you like. Now, what we know is that the human central nervous system pericytes differ from pericytes around blood vessels in all the tissues of the body. The importance of that is the CNS pericyte, the CNS pericyte is very much involved in maintaining what we call the blood-brain barrier. This is the barrier that occurs only in the central nervous system blood vessels that regulates what goes into your brain and, if you like, helps get stuff out. So the pericyte is very important in maintaining the integrity of that blood-brain barrier. It regulates the flow to the brain, scar formation, inflammation, so a very important function. And leaky blood-brain barrier, particularly with aging and everything, has been associated with many CNS diseases. They also are involved in the transfer of proteins between nerves. We often thought that nerves were nerve-nerve connections, but there's a thing called nanotubules, little, if you like, tubules that can connect nerve to nerve. And of course, this allows them to transfer proteins and metabolites, and the integrity of that's very important. If that goes wrong and it starts transmitting faulty, misfolded proteins, and two that we do know of is alpha-synuclein in Parkinson's disease and tau protein in Alzheimer's. If they get transferred from cell to cell through these tubules, then the diseases spread. And that's a very active research topic at the moment, is what maintains the integrity of the nanotubule. We work with Richard Fall and Mike Dragonoff's group here, and this is presentations up on the ASX, so if you want to read more about them and their importance, you'll note that that's a very topical paper having been released this year. So that is a very hot topic that you address. So clearly, there's an unmet need in the art for enhancing CNS pericyte health. What, if you like, can protect them from damage and enable them to maintain the integrity of the blood-brain barrier and carry out their anti-inflammatory, autoimmune, and hopefully removing the rubbish from the brain. So we initiated the project that we announced, and the first part of that project carried over the last 6 months. We have found that our NT cell does protect human pericytes. Now, the important thing here is that we're using human tissue samples that are obtained from patients who have no disease. In this case here, that one is Parkinson's disease and this one is Alzheimer's disease. Now, if these pericytes are dying, that's 100% cell death up here. So you use an agent, in this case peroxide, to cause 100% cell death. If you incubate with the NT cell, you can prevent that death occurring, if you like. So the capsule is the blue line here, and as you can see, it has moved a huge amount. Apologies. It has moved a huge amount over this way, and that's a very potent function, and certainly from the Centre for Brain Research, it's the first time they've seen anything that can do that. Now, that's the capsule itself that encloses the choroid plexus cells. If we take the media, just where the cells are releasing things into the media, we virtually get the same thing happening. So something's coming out of NT cells that's protecting pericytes. If we do that, that's in Parkinson's patients, Alzheimer's patients, normal patients, the same thing is occurring. There's a protection of human pericyte function. So that gives us a bioassay, if you like, to identify what that is. So we protect the pericytes in human tissue cultures. Very few places in the world have access to them. The media has the activity indicating there's an agent there, and the effect is specific to choroid plexus cells. If we try other cells, nothing happens. It's something coming out of choroid plexus cells. So our next step here is to identify what it is, and if we can synthesize it and find a protective agent, then you have, if you like, a potential treatment, and that would not be a transplantable one. It could lead to an oral formulation, injectable or something like that. The person who can do that is probably one of the best medicinal chemists in New Zealand and Australia, and that's Margaret Brimble, and we work with her group in the next phase of this project to, first of all, identify what it is, and hopefully identify the peptide fragment that's important, and if we can synthesize that, you would have a very attractive product candidate. Patents. With this result, we filed the second patent, which is pericyte protective agents for neurological disorders, including neurodegeneration, and that's a rather broad patent that we do have. The prior patent is important because, as a general patent, that covers many areas, which includes the cell-based therapy areas that we might look at, which included the retinal degeneration and the chronic ear cochlear protection. So our next steps is to confirm what the lead compounds are, and that is to understand, probably using the criteria of what investment we can make to take one of these projects to a fundable milestone. So they will compete with each other, and we hope to address that as soon as December with another board strategy meeting. And also at that time, we will have the data from the follow-up on the Parkinson's patients trial to see, is there any lead in there that 1 year later gives us a target to see if we can meet the criteria put to us by the Medicines Control Agency here. So if we succeed in all of this, there's only one measure of that, and that is shareholder value. Obviously, whatever we do, we get data, we'll announce that. We've got quite a good history of reaching milestones. Unfortunately, the milestone was a big one that we did not get the clinical endpoint in, but I'm confident that we can do this and then hopefully get a milestone of here is a product candidate that can have the ability, with the funding that we've got, get to a fundable milestone. So with that, what I'll do now is introduce you to Dr. Barry Snow, the principal investigator, and I'll let him just talk a little bit more about the clinical trial, and then after that, be very happy to take questions.
Afternoon, everyone. So I represent the clinical team who did the research for this study. I was the primary investigator, so helped with the study design, took responsibility for the study, which I do, and during the study I was part of the blinded team. So there was the neurosurgical team and the radiologist who knew absolutely what the patients got, and then there was the blinded side, which included the patients and the assessing team, and I was part of that. So I found out the results at the end of last week, just like you did. Yes, we are disappointed. We're driven by something a little bit different to investors. We're driven by a need to make a difference for our Parkinson's patients. And of course, so were the patients. And now, when we recruited patients into these studies, we have to get their consent to have treatment that's unregistered and experimental. And we use a concept of expect versus hope. So we tell the patients that we really hope this works, and so do they, and we think that's healthy. But we also tell our patients that if they expect it works, that they weren't welcome to join the trial because of the possibility of being disappointed and left out. And it's something that we work quite hard in our consent process, because people have to go into this and just contemplate for a moment somebody drilling a hole in your head and implanting a foreign tissue that is not proven to work. And so that required quite a lot of careful, informed consent to make that work properly. About this point, the patients would sort of ask, well, does it work? And our answer always, well, if we knew it worked, we wouldn't do the study. So that's the background for the patients, and of course the background for us. And I know you're disappointed, and if you're an investor, well, you took the chance. But don't forget, so do the patients as well. We've talked to all of our patients. It's interesting that we've had a universal response, which was worth a go, that they feel proud to do it, and they feel very well looked after during the process. So certainly none of them have expressed frustration or anger at the outcome of the study. So, and I know that you're mostly interested in the bottom line, and the bottom line is what we set out to prove, which doesn't refer all to that second lot of data. So when you do a research study like this, it's a little bit like the rules of a sporting game. You know, you only get 1 point if you kick the goal between those two posts. And so when you set out in a study like this, you have to define an outcome that you're aiming for, and the outcome was a statistically significant result in the motor score of this thing, the Unified Parkinson's Disease Rating Scale, which is the absolute standard score for measuring these sorts of studies. And so what this study was based on, it was positive or negative if we got a statistically significant result on that outcome. We were guided to believe that was a reasonable approach looking at the phase 1 study, but in fact we didn't get that. What you do now with a research study like this is you pause and you dive back into the data and you look at it in different ways, and that's what we're doing now and next, and that will actually take quite some time as you kind of rotate that Rubik cube in your hand to look at the data. So there'll be more to say, but same answers before. If we knew the answer before we went in, we wouldn't have to ask the question. I'm prepared to take more questions about the study. I can talk about the safety and other efficacy variables. I've got a question.
My name's Ryan Smart, I'm a shareholder.
Hello, Brian.
The question is, when you say it will take time to analyse the data, what is the time frame to analyse the data?
I need some guidance from these folk here. The way it works in science is that you you run your statistics, then you look at the data, and then you so-called pivot the results, and then you look at it again. And in a normal kind of scientific level, that sort of process takes weeks, days and weeks, but not months or years. It's that sort of time. But I know you've got different rules about share market reporting and things like that somebody here on the board might be able to answer better. But it's that sort of time that will actually match pretty closely to the amount of time before we get the 12-month data on that, the 80 group.
I'm Steve Skinner. I work for LCT.
Indeed. Hello, Steve. Nice to meet you.
So if in this coming time you have some positive data, will that allow you to present it to the ethics committee and ask for more patients? And if you can, do you have enough money?
Okay, well, that's not a question for me. Yeah.
The answer, Steve, I think is yes. We have, Barry mentioned analysing the data that we have, but remember these patients are also being followed by a extended protocol, so we go out 1.5 years, 2 years, so we get more data. If we get anything that is, if you like, mid-safe enabling, yes, we would talk to them immediately about that. MedSafe were very helpful in designing the protocol, if you like, so they know exactly what the endpoints are that we're talking about. And if we do see anything that reaches a statistical significance, because if you're going for any regulatory approval, you can plead for mercy on the clinical need, but they operate on statistical significance. Now, if that can be achieved by a feasible study within the confines of patient availability, it's that Barry can comment on, but also within the finances that we have, yeah, we would do that. So it's data-driven. The answer is if we get the data, of course we would do that. And as I mentioned, probably our next review of that data would be in December of this year.
Paul.
That was a very neat study. And I guess the surprise is not all that surprise. It relates to the control group who, as you said, the trial showed a universal improvement. From a clinical point of view, how long do you think you expect a placebo response to continue for? And in wanting to examine future data, if the placebo response were to fade, the question still remains. Is the improvement that you've seen in your patients of clinical value?
Sure. So there's a number of questions nested in there. So we are using the proxy for clinical improvement of the UPDRS, and an improvement that translates to a change in the person's life is about 5 points. That's reflected further in the PDQ-39, which is one of our other outcome variables, which is a quality of life score. And interestingly, there was a fairly substantial improvement in that 80 group as well. So, you know, if you're an optimist, you look at that and really wonder if there was some improvement there. How long does the placebo effect last? The general expectation is about 6 months. And then other studies, it tends to drift back there. But it's highly variable, and you can see different amounts in different studies. So in some studies, you see 0 placebo effect in Parkinson's, and others you see what looks like a sustained effect. And probably those sustained effects are reflecting different things. The thing about studies like these is that patients get an intense amount of attention. And when they get intense attention, they do all sorts of other good things, like they feel more comfortable, they sleep better, they're more likely to exercise, they take their pills. All the stuff we're telling them to do in the clinic all the time, that finally they seem to have got motivation to do it. And so I don't think although that's the placebo group that improved, it's more subtle than a pure, quote, placebo effect. It's probably a mixture of that attention from us to the disease and the patients to their self-care as well. But that does provide the baseline that you have to measure everything else against. So it's a complicated answer, but it's a complicated world, the placebo effect. It's what drives the whole alternative medicine industry, of course. Yeah.
Yeah, but I just want to understand, you are assuming that the placebo will always be And assuming that placebo may stay to some extent, the point is, do you have a viable treatment, a worthwhile treatment, if you see an improvement of the order that you have observed with the AG group?
Probably.
And therefore you're asking us to actually sit and wait before a final conclusion can be made for the enormous work that you've done in this trial.
So a worthwhile, when you ask about how much difference is a treatment worth doing, that depends where it sits in the hierarchy of a treatment. So, for example, if it's a very simple pill that's no effort to take with almost no side effects, you would tolerate a very small improvement in return for that minimal inconvenience. Whereas if it's something huge, like, say, deep brain stimulation for Parkinson's, where probes are left in the brain, there's wires to a pacemaker, you'd expect a 20-point improvement in the UPDRS to make that worthwhile. For a treatment like this, which you would do relatively early in the course of the disease, which is actually quite a simple neurosurgical procedure, which requires no ongoing maintenance, and it doesn't prevent the patient getting other treatments in the future, anything on 10 points or so would be, in my view, a reasonable trade-off to take. And that's the sort of numbers that we have been seeing if you believe the results. So if you go back to the first group of patients, and I'm the first to agree that we saw a different result to this group, but the amount of improvement we saw in that first group of patients would make it a viable treatment.
The 80 capsules, the brown one that you had that came down under or reduced under the placebo itself.
Yep.
Would you be able to make a statement that that in itself would be enough to halt the progression of Parkinson's? Without any benefits, just the halt?
You can't say anything other than what the data says. So, firstly, you have to say, was that a real effect? And if you pick and choose, you can always show something that is believable. That's called fooling yourself, and you have to be really. That's why we do statistics, to stop fooling ourselves. But you're asking a hypothetical question: is that if that brown line were a real effect, what you're not seeing there is halting. You're seeing an improvement, which is actually the biological hypothesis here. The hypothesis is that you're encouraging resprouting of those neurons, just what you see in the animal models, and the hypothesis is here. So the hypothesis we went into here was not to hold the disease, but was to improve the disease.
But there is a possibility from the results that you've currently got, from the baseline being 0, that the green line and the brown line have reduced the impact of Parkinson's on the patient. May not have stopped it, but reduced.
I'm just going to repeat myself. Statistically, there's no difference, so you can't say that. If you want to call that a trend and hope that it becomes positive, yeah, you can say that. But that you'd be kicked out of the park in any scientific presentation.
What might be helped here is that that's what you're testing in the long-term follow-up. Does that get to a statistical significant point? And that's the beauty of the follow-up. So there's a hint there. It's not statistically significant. You have to announce the trial endpoint, which was not met on a statistical significance, but you might say exactly what you're saying, Ryan, justifies the follow-up to see if we can get to a clearer picture.
Just one question on the AG group. Was the variation that you showed, say, on the was it wider or more compact on that group, just out of interest, given that that's the
Very similar amount of variation across the groups.
yeah.
But that's what you expect in biology. Yeah.
That's what I was thinking.
And my other question relates to Immupel. Is that directed at you?
Yep.
Okay. Is there a possibility of licensing Immupel to other cell therapy providers around the world as a delivery system?
The Immupel technology is quite well known. There's a lot of publications on it, and I think it's got to the point now where your trade secret in production is probably more important to what you have as a patent. So are trade secrets sellable, to who, and things like that? There are inquiries, but in terms of it being a financial big returner when the patents are virtually expired, it really is linked to the trade secret, knowing how you do make it. And I might say there are. The group here at LCT, I think, have an incredible knowledge about this, and they can make things to specifications. So maybe some value, but not from could you make a large amount of money from an out-license of that technology.
With the amount of other cell therapy providers around the world, is it difficult to encapsulate their technology inside your InfuPal technology, or is it impossible?
No, I think the different groups, depending on the cell they're doing, are encapsulating, if you like, and more their bigger issues, I think, relate to does that give them immune protection for what they're looking at, and what are they trying to protect against, if you like. But yes, there's a lot of experiments to try and improve that. The technology we've used has been known for quite a while, and I think it's been perfected at LCT for encapsulating choroid plexus. Is that appropriate for somebody else and where they're at? That I think depends totally on their target and what the encapsulation is supposed to do.
I think InfuPal's a market leader. It's still the only delivery system in the world that doesn't require the patient's immune system to be lowered. Whereas other delivery systems that are being put, they're lowering the patient's immune system, and that's of no benefit if they have pre-existing medical disorders. Whereas your delivery system can be injected directly into the body. You've got no autoimmune system to kill it off. I think the technology is extremely valuable. I think it's been overlooked. I think it's the best tool in the entire kit.
I get Bob Elliott, who's been using the technology for a long time.
I'm posing a comment as a shareholder rather than a board member here.
Thank you.
And indeed the one who dreamt up the idea in the first place. When we set out back in 2001 with this whole idea, we were operating on the idea, and your question over here is really relevant to that. We might be able to hold the disease progressing. That's what we were aiming at. Which is Alzheimer's now in my mind, but we'll switch to Parkinson's later. We had no idea that the cells, the NT cell, might actually reverse the disease. And the first person to come up with evidence that it might, sitting over here, Steve Skinner, showed this in rats, which is a sort of a model of Parkinson's was produced, and also in monkeys, that actually saw new cells coming from nowhere and being regenerating these dead cells in the affected part of the brain. And we got terribly excited about this. This was a regenerating brain. A damaged brain was actually growing again. The only problem was these were very young animals. The rats and monkeys were young. And what we were seeing there may well not occur in the aged brain. I have good personal evidence of that. So your question is right. Would it be worthwhile just stopping the disease in its tracks? Yes. But this trial was not designed to show that. It could show with longer follow-up that that's exactly what's happened, but we can't say that at the moment. The trial failed on those conditions we laid out. The rules of the game were 6 months follow-up, statistical significance on those features. It didn't happen. It might happen in 12 months, 24 months, 2-3 years, who's to say? We will follow it. That is the intention of the company, follow it, and for that period of time, to cover exactly what you're saying. I think it's a very good comment. It would be worthwhile if I had Parkinson's disease at an early enough stage. I would want it stopped. It'd be nice if I reversed it, but if I stopped it, that'd be good enough for me.
Do you think the bar was set a little high, trying to get reversal?
Yeah, we got a bit enthusiastic.
Well, look, if there's no other questions at the moment, I know... Sorry, there is one.
I've been involved with these LCT since the first IPO. I've participated in every placement, invested online on the market as well. I'm concerned with all this that there is no question about the science. There's no question about the ability of the board and what have you as it relates to that science. But we're not a philanthropic society. We want to help people who are disadvantaged, we want to help, but there has to be a bottom line somewhere in this. We're investing a lot of money. I'm well into the 7 figures in this company, or was. And as a consequence, nothing here has told me that there's going to be a return to the shareholders. Maybe the never never. I'm not sure I can go another never. Can you give us a feel of the value of some of these things so that we should stay in, we should participate in new raisings if needed, so that we know that there is an end game here, but there just seems to go on and on and on. 13 years so far.
And also that the gentleman's comment is reflected across all of the share trading forums over the past week. Every single one, United-based, UK, Australian, everyone's asking that question. Where is the return? Where are the dollar amounts next to value of what these treatments will be worth?
So we have to accept those comments and criticisms. I haven't been with the company as long as you certainly have, and going into the future, so why should you hold your shares? Well, that's obviously an investment in venture capital type decisions. There's risks and you have to assess the benefits, and that's right. So where are we going? I think of the cell-based therapies, we're still trying to, one, see can we get anything further out of this study, and I think you've heard some good rationale, and Bob has eloquently indicated that if we can just stop the disease and with long-term follow-up get some evidence for that, then that's one thing we're definitely doing. With the cell-based therapies, they're based on, we know that technology, so we know the product, we can GMP make it, and we've got targets that are very close to once again addressing a clinical trial. We have to do that carefully because we wouldn't want to do that and then disappoint you again, and we certainly wouldn't expect you to want to contribute funds for that. So we have to get there with the money we've got. The third one, which is why I put up the pericyte protective agent, is that can take you down the track to where you can get a synthetic chemical product. And if you're going to go for Alzheimer's or a heavyweight hit like that, then that's an attractive thing to go for. Why is it attractive? Because if you're synthesizing a chemical, that can lead to a tablet and you can take it orally. So the X is obvious. You prove that, and then I guess I spent 40 years of my life in the big pharmaceutical industry. You're talking their language. That's the stuff they go for. So in that situation, LCT would only get to proof of principle with good patents on compounds, and your exit is big pharma going after anything that has the word Alzheimer's, patents, Parkinson's, but they would do probably the phase 2, 3. That is their language, and most of those deals they take over and they do it professionally, and you're approaching a global market. So you've got 3 bets that you're going to make your call on. Are you going to hold your shares? 1 is the follow-up, Parkinson's. 2 is the cell-based therapies. We know that, and we can make the stuff. And we're talking in an advanced way with 2 interested parties who can do the clinical. And third is the innovation in an area where pericytes, nanotubules, synthetic chemistry, patents, if you like, you're talking big pharmaceutical company exit there. So those are the 3 things. But what we do know is we have to get to an endpoint with the money we've got. We can't come back to you and ask for more until we do that.
No chance of another JV with Big Pharma? Have you been approached by any Big Pharma with interest to the current results?
Big Pharma operate off product and data, and they're prepared to do the phase 3, if you like. Cell therapy is a bit limiting with those that do have interest, and those that do tend to maybe look at stem cells rather than xeno, but that's just the way they are. They all talk a medicinal chemistry product. They understand all of that. And that to them is easy because you can synthesize it, cost of goods are less, and you can do your big phase 3 and market to the world. So that's a different language altogether. So really it's between those three things that I think you should try and make your decision. But as a company, total sympathy with you. We thought we were targeting something that we did a lot of homework on, design of trial, international experts, wanted a result. If we could get to one, as Bob says, that halts the progression, yeah, we've achieved a heck of a lot with that. But we do have these two other approaches, and the funding we have, which of course we raised for the continuation of a positive effect to go forward to a product approval, that money now has to be applied to one of these three areas, and we have to get to a fundable milestone as an outcome.
Are you able to update us on the diver cell?
Maybe if I pass you to the chairman of Dole, we do have this joint venture with Otsuka Pharmaceutical Factory, and our job as LCT is to support them. The joint venture's Diatrans Otsuka Limited, and the chairperson is Bob Elliott. So Bob, do you want to come up here to the microphone and...
Yes, well, as previously explained, we went to a joint venture with Otsuka Pharmaceutical Factory some years ago, and we conducted clinical trials through that joint venture in both in Argentina, both phase 2 studies, and the decision was made at that point not to proceed with that product at that time. The results of those 2 studies, one has been published, Dr. Hashimoto, who's sitting over there, is the primary author, and the other one has been published on a government clinical trials site recently. The results were both showed a distinct improvement in the control of blood glucose, but no cure for diabetes. Both the blood glucose stopped dangerous variations in blood glucose, and that would have been a big advance in the treatment of diabetes, but it did not constitute a cure. Our colleagues in the Otsuka Pharmaceutical Factory decided some modification of the product was needed, and they have proceeded to do that through a licence granted to them by the joint venture company in the United States of America, and they are currently seeking FDA approval to proceed with the clinical trial of the modified, hopefully improved product. Now, we get regular updates of where they're at. Like us, they're finding progress is a lot slower than you'd like it to be. The regulatory pathway in the United States is more difficult than it's been here in New Zealand. In fact, dare I say it, the only country to grant regulatory approval for the diabetes product, for that matter, any product based on living pig cells, is New Zealand. So they face many of the uphill battles we face. So they are making progress slower than they would like, certainly slower than we would like. But yes, it's in hand, things are happening. We're not doing anything here in New Zealand. Otsuka Pharmaceutical Factory are sinking huge amounts of money into progressing this, the sort of amounts of money we would not be able to get in New Zealand. I'm talking in scores of millions of dollars, probably more. But that's where we're at the moment. Yes, the product is being approved, and the intention is to try it with FDA approval in the United States. If it is approved, then OPF would grant us a licence free of charge for the rest of the world outside the United States and Japan. Question answered, I hope.
Yes. Richard LeGrice, I'm a shareholder. Look, I totally understand that by investing in this company, you're investing in a company that's very high risk. And I appreciate the fact that there are some extraordinary clever people in this room that are involved around what's going on. But I would suggest that as a shareholder, I'm lacking a clear strategic plan. You've got this going on here, this going on over here, and sitting here right now, I don't quite understand what the company's vision is, what its focus on, if it goes down a road that's a dead end, when will it decide that, etc., etc. And I think this is really important going forward from here because, to your point earlier, you've got $6 million left. With a share price at 3 cents or whatever it is, it's going to be impossible for the business to raise funds. And so, yes, as a shareholder, I'd like to firstly maybe get a comment from you on that, but also feel it's imperative for the future of the business.
And I think that's fair comment, so we accept that. I think what we have to do is we are a science-driven company, so we have to go on the data we generate. Now, the next step in generating data is in December. The board has rescheduled another board strategy meeting there when we do have data. That data is going to determine from the projects that I've mentioned which data supports the further investment from the money we've got that has got to take the company forward, if you like, to what I would say is I call it a milestone generation, whether it's a fundraising generation or something like that. The time frame is urgent, and I agree with you on that. But if we tried to make the call now and roll the dice, I think that would be wrong. We need to get more data on the Parkinson's. We need to look at the current projects from what the next step is and understand the funding. Then there's a certain competition from what we know in December from which we have to make a call on the way going forward. So I agree totally with you. It's an urgent thing, but if we tried to do it right now, reacting immediately to results last week.
That's the last thing I suggest.
Yeah.
I mean, I'll also say, obviously, Dr. Elliott commented on the partnership with our Japanese friends and the huge amounts of money that's being invested in it. I don't actually understand that, and I don't understand as a shareholder what that's going to mean to me. If it's successful, what sort of return on investment is the business going to get? What revenue is it going to generate? Is there any targeted budget around it? I guess these are sort of things that I think that I would have much more confidence in reinvesting in the business if I understood some of the other things much better that the business is involved in. Because it's not just about this one trial, in my opinion.
Well, thanks for that. I would agree with that, and I think strategy meetings with the OPF partners, Dole, there's meetings tomorrow, if you like, but once again, we're getting more data on where we're going, and then can we present to you shareholders a rational strategy of where the investment of the funds that we have are going, if you like, based on data.
Thank you. I've said enough.
No, thank you.
My final question is, why does LCT shy away from paid investor content or paid investor awareness? Out of the 300 biotechs that I sort of look at, 200 of them are constantly sending out stuff new through paid investor content channels and broad stroking them. One of them alone has a 200,000 share traders on their database, and yet I don't think I've ever received anything to encourage me to invest into LCT. So you've currently got a share pool of about 3,000 that are domesticated between Australia, North America, New Zealand and Japan. Whereas I think if you had had a much larger broad stroke shareholder base of 30 to 50,000, you wouldn't have had the knee-jerk reaction that you had on Friday. And let's face it, on Friday it's got to be said, $120 million was wiped off the market cap. It went from $141 million to $21 million on Friday afternoon. That's a colossal amount for any company. And I think at heart, and I respect all of you for coming today, absolutely, and you've answered all of my questions. But at the end of the day, you're a business. You're Living Cell Technologies Limited. You're a business. You're not a research institute. So that gentleman's question in his comment before is correct. There's no hard numbers next to this. Divacell will revenue you $100 million. NT Cell has the possibility of revenue $200 million. And I liken it to almost the Tesla model of saying we're going to produce all these cars, but we're not going to sell one until the first one comes off the line. You know, and so consequently, I need to understand why you've avoided that. I understand you're science-based and science-driven, but so are about 200 other biotechs that constantly send out information trying to gather up new shareholders, increase their base, increase awareness of the company. And it's not just about shareholders. You could have had third-party relationships generated from that investor awareness. You know, you only need one email to go out to North America somewhere and suddenly you've got the Mayo Clinic knocking on your door, or some unknown clinic, or unknown investor. Again, 7 to 8 digits, you know? And I just can't comprehend that. I understand you're science-driven. I understand you want to make bold statements that are supported by the science, but there's so much more to LCT. You've got longevity in the marketplace. You've got credibility. You've received government grants. All of those things distinguish you from your competitors, right? And you have competitors. The competitors are trying to get funds from us here to forward their research. So I need an understanding of why the PR is being bagged, why there's been no investor awareness and paid investor content.
Okay, so we do have a PR program that we do. We have a PR agency that we do disseminate information from. The key to biotech PR is only 2 things: product and data. And if you don't have product and data, you're hyping bullshit. A lot of biotechs do that. They spend a lot of money doing it, releasing every month. And what is it? It's not going to get you to a product. You mentioned Tesla. They've got a product. We're looking for a product. If we get a product, we'll hype it. Are we aware of the international scene? I go to international meetings ranging from JP Morgan to a lot of colleagues I have in the industry, and they're fully aware of what we're doing. They know we were going to get a result. They know exactly what we're doing, but it's still about product and data. And if you're trying to make it anything else, I think you'd be here accusing us of deceiving you. And you can read in the newspapers lots of hypey stuff. Whatever happened to that product that was supposed to cure something? Why is there no cure for Parkinson's, Alzheimer's? About every week in the media you'll read something. What is that achieving? So it's product and data. We like to pride ourselves that when we do have data, we do release it. We have to by ASX regulation, but we don't overhype because I think that's misleading you and that's not telling the truth.
Just one question. Are the board going to stay on? There's no one's disappointed enough to step back and say I don't want to do this, because I get to a lot of AGMs, and there's not very many that you go to that the directors have a lot of intellectual horsepower, articulate, understand the subject that you're talking about inside out, and have skin in the game. So I would be reassured if the directors did plan to stay on.
And have skin in the game. The chairman, the CEO, and a director have no shares. I don't understand that.
Well, I'll come in from the CEO. With the first time I've been a CEO for an ASX listed company, their rules about disclosure and everything are quite onerous, and because of that, while I might have wanted to buy shares, managing it's a lot easier to declare that you're independent and you don't hold them. You have options, of course, so I'll lose all my options here, but in terms of, if you're looking for a reason why isn't the CEO bought shares because he doesn't believe in that, there's nothing to find there. It's more linked to if you're a good CEO, you should be moving the company down the track. That's me, but I'll leave the director's question to.
I lost my deposit on my retirement card as well as you, and I had significant disaster in there too. I lost that as well, so I understand your point of view very well. So,
We have the results from the poll. I can tell you 2 of the directors have been re-elected. Sorry. I was just commenting that we have the results of the poll, and 2 of the directors who were put up for re-election have been re-elected. So all the resolutions that were put before have been passed, largely in the order of about 93-94% approval. Are there any other questions that anybody would like to put forward? If not, can I...
For some of the shareholders that aren't here, I'd just like to thank the gentleman for coming today. It was a big pill to swallow, and you came along and faced up to all of the questions, and it's a credit to you all, and a credit to your organisation.
Thank you very much. Appreciate that. So I think there might be some refreshments over there, and if you wish to talk further with us individually, we're welcome to engage further with you. Thank you very much for your attendance.